Hello everyone. We are pleased to have the opportunity to present our work, which focuses on the temporal response patterns of human gut microbiota to dietary fiber. The gut microbiota is a highly dynamic and complex ecosystem, but its temporal responses to dietary fiber are not yet fully understood. In this study, 19 participants first underwent 14 days of normal diet, followed by 14 days of dietary fiber intervention. Using guild-based and time series approaches, we identified distinct microbial response patterns. By integrating multi-omics data with time-delayed analysis, we further identified key microbes and metabolites linked to improvements in host metabolism. In summary, the highlights of this study include capturing dynamic microbial shifts, uncovering stable interactions among co-occurring microbial members, and identifying key microbiota metabolite associations linked to host health. The clinical study design is shown in the diagram, and fecal samples and continuous glucose monitoring data were collected daily during both the observation and intervention phases. At the end of the study, 10 participants in the overweight group and 9 in the T2D group completed the trial. Clinical assessments showed that, during the intervention, the percentage of time per day with glucose levels between 70 and 180 mg per deciliter remained above 70% in both groups, with no significant changes from the normal diet phase. Meanwhile, dietary fiber supplementation reduced glycemic variability and fasting plasma glucose across both groups. We performed shotgun metagenomic sequencing and assembled 1,100 high-quality metagenome-assembled genomes. Dietary fiber increased microbial alpha diversity in both groups, with significant changes observed within the first 3 days of intervention. Additionally, daily sampling revealed that changes in gut microbe composition began as early as the second day of intervention. We partitioned co-abundance groups, abbreviated as CAG, based on stable intermicrobial relationships. Based on daily sampling data, linear mixed models revealed a substantially larger number of CAGs with significant abundance changes during the fiber intervention compared to 2-point comparisons. We further identified 8 temporal patterns of microbial changes in the overweight group. Functional gene analysis showed that CAGs sharing similar abundance trends also exhibited consistent functional traits related to carbohydrate metabolism and transport, aligning with their taxonomic similarities. Similar results were also observed in the T2D group. Both groups exhibited significant fecal metabolome shifts during intervention phases, with changes detectable as early as the first day of the intervention. Pathway enrichment analysis of upregulated metabolites indicated enhanced amino acid metabolism during the low-dose phase in the majority of participants. Moreover, high intervention led to a broader range of upregulated pathways, particularly those related to phenylalanine and tryptophan metabolism. Using local similarity analysis, we further investigated the time-delayed relationships between microbiota and metabolites. In the overweight group, CAG80 was positively correlated with 4-pyridoxic acid, with the microbial shifts preceding metabolite changes. Furthermore, ALK-MAGs within CAG80 harbored encoding enzymes essential for 4-pyridoxic acid metabolism. In addition, 4-pyridoxic acid levels were significantly negatively correlated with multiple glycemic indicators. We conducted the same analysis in the T2D group and also identified key CAGs and their associated metabolites. Results suggest that dietary fiber-responsive gut microbiota members may contribute to improved glycemic control by modulating specific metabolites. Furthermore, mediation analysis identified several significant linkages in both groups, providing insights into potential interactions among microbiota, serum metabolites, and host metabolism. In summary, our study highlights the value of high-frequency sampling in capturing dynamic microbiota responses. Moreover, by linking microbiota and metabolites associated with host health, this approach provides potential targets for causal research and clinical applications. Looking ahead, future research could its application across various diseases to deepen our understanding of microbiota dynamics. Thank you for your attention.